Hi guys, this is video 803. We're going to talk about organelles and specifically the ones that are used in protein synthesis. So the whole goal of this chapter is to look at how proteins are formed. And quite a few of the organelles we haven't talked about yet because, again, we were waiting until we talked about that application. So let's label the ones that we have talked about. The first one that we talked about was the cell membrane. So I'm not quite sure which, yes, let's put cell membrane over here. And we did a lot of detail on that. We talked about how substances get into and out of a cell. Then we talked about the nucleus. That is where the genetic material is maintained. And here we see chromatin. So you see the loose chromatin. Because remember, it doesn't condense into chromosomes until we undergo meiosis. And meiosis, these guys over here are the centrioles. Centrioles. That's an R. And these are used in meiosis during the cell division. Then we went on to cell respiration and photosynthesis. So we know about the mitochondria. And we also talked about these little suicide sac guys called lysosomes. Lysosomes. And then we have these structures here that lie underneath the cell membrane. And I should have done this when I did the cell membrane, but we talked about this back in the cell membrane chapter. These are microfilaments. The other thing I forgot to say is that if you guys are looking at a black and white copy, take a second and let's color code that. So pause the video here and go color code your notes. All right, welcome back. So what we're going to be talking about today is this structure, this, and these little dots. So let's start off with these little dots. So you see one is pointed to here, and there's an arrow coming out. We also have them on this orange structure, and these are called ribosomes. And I'm going to switch and go to colored pens now. I'll kind of color code. Well, these guys are in black, so I'm going to keep this in black. No, I'm going to do red. I lied. So these are ribosomes. And they're very small. Everybody has ribosomes, even prokaryotes. So it's one of the few structures that you find in both eukaryotes and prokaryotes. So what do I want to say about ribosomes? They're found in all organisms. Found in all organisms. And they are the site of protein formation. So when we form proteins, it's going to happen on the ribosomes. And again, we have free floating. So here you see a free floating ribosome. Oh yeah, that reminds me, cytoplasm. That's another one we talked about. Excuse me. And then some of the ribosomes are attached to this structure. And this structure is called the rough endoplasmic reticulum. Rough endoplasmic. endoplasmic reticulum. And it's rough because it has ribosomes. So the rough is due to ribosomes. So rough is due to the presence of ribosomes. And what's going to happen is in this structure, the proteins are going to form on the ribosome, and then they're going to feed down into this network, and they're going to pass back and forth and back and forth. They're going to go this way, and they're going to go this way, and they're going to go this way, and they're going to go this way. And eventually they're going to pass into this purple structure. In purple, we have the smooth endoplasmic reticulum. Smooth endoplasmic reticulum. So we haven't really talked about what the rough endoplasmic reticulum and the smooth endoplasmic reticulum actually do. So what we're going to see here is that protein formation is going to take place in the ribosomes, and then the proteins are going to move into this network. They're going to move back and forth and back and forth. And in both the rough endoplasmic reticulum and in the smooth endoplasmic reticulum, so for both of these, the proteins are going to have folding and packaging. Folding and finishing, I guess you should say. So when they come off the ribosomes, they're just this long strain of amino acids. Remember, the monomer of proteins are amino acids. And so they have to get folded up, and that's going to take place here, and then they get finished when they get into the smooth endoplasmic reticulum. In addition to finishing proteins, the smooth endoplasmic reticulum will also finish fats. It'll also finish lipids. So there's some lipid repackaging that takes place on that smooth endoplasmic reticulum. And that just leaves this little area inside the nucleus. This is the nucleolus. And this guy creates the pieces of ribosomes. So this forms the ribosomes, basically. They get assembled out in the cytoplasm. All right, so that leaves this guy here, and I'm running out of space. So let's see, where do I go with this? I'm gonna do a big arrow down here, and I'm gonna do this in pink because this is kind of a light purple. This is called the Golgi Bodies or the Golgi apparatus. Depending on how old the resources you're looking at, some of them will say Golgi Bodies and some will say Golgi apparatus. And the job of these is to package and ship the proteins out of the cell. So they come off the smooth endoplasmic reticulum and they go into a vesicle, and they go into these stacks of vesicles. So you've got layer 1, layer 2, layer 3, layer 4, layer 5. You get these stacks, and the proteins then kind of, the stacks rotate, and then this top stack will come out to the edge of the cell, and it'll fuse with the membrane, and it'll release its contents through exocytosis. But the Golgi bodies, their function is to package and ship proteins out of the cell. Ship proteins out of the cell. So our new ones today, let's go over those. Let's go back through all of them. So we've got in chapter, it was like 2 or 3, we had the cell membrane, and we also talked about there the cytoplasm and the microfilaments. So, you know, if you don't remember, this controls what goes in and out of the cell. This helps support the cell. This fills the cell. Then we went on to, we also talked about the nucleus early on when we talked about prokaryotes and eukaryotes. Then we went on to cell respiration and photosynthesis. Mitochondria, they produce ATP, powerhouse of the cell. Somewhere in there, we talked about lysosomes. They're the suicide sacs. When they open up, they can destroy the cell through apoptosis, or they can break down organelles. Then we had the centrioles in the meiosis chapter. Those guys are responsible. They attach the spindle fibers to the chromosomes and pull everything apart. So our new stuff for today, we've got the nucleolus that's forming the ribosomes. We've got ribosomes floating around in the cytoplasm or attached to this endoplasmic reticulum, and they are going to form the proteins. So site of protein formation, very important. The proteins then get folded and finished as they move through the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Then they're going to bubble off the end and come down into the Golgi apparatus, where they're going to get sent out of the cell. So the whole point is to send the proteins out of the cell here. And if you remember back in, it was like the 2nd chapter where we talked about the categories of macromolecules. There were, we had the nucleus and it had the DNA in it, and we had the carbohydrates going into the mitochondria. But if you remember, there was an assembly line, and that was the protein manufacture the formation of proteins, and we had the amino acids going in, and it was like a conveyor belt that went through the cell. It's actually pretty close to the truth. So we start to form those proteins and we send them through this conveyor belt, and then the conveyor belt kind of continues and takes the proteins out of the cell. So you can kind of think of this as ribosomes, and then we've got the pathway for the proteins to get sent out. Of course, not all of them get sent out. Some of them are used by the cell, but a lot of them do leave. Okay, so that is a review of old organelles, and that finishes up all the new organelles that we need. So now we're ready to start talking about transcription and translation. On to the next video. Thanks for watching.